Okay, thank you, Dr. Samar.
Okay.
Bismillahirrahmanirrahim. Assalamu alaikum and good day, ladies and gentlemen. I'm Hazima Mozam and I welcome you all to COMSTECH NDT joint lecture series. This lecture will be delivered by Professor Dr. Carlos Robello. The title of his talk today is Genome Organization of the Triclosan complex. Again, I would like to highlight here that the purpose of this COMSTECH NDT lecture series is basically to sort of create an awareness among the researchers regarding the tropical diseases around the world and to elucidate their prophylactic and diagnostic sides of the research. With this, I would like to introduce our guest speaker for today. Professor Dr. Carlos Robello is a professor of biochemistry at Faculty of Medicine, Universidad de la República Uruguay. He is also the principal investigator at the Institute Pasteur de Montevideo. His research is focused on the study of the biology of Triclosan and its interaction with the hosts, using molecular biology, genomic approaches to better understand the parasitic lifestyle and the infection and identify new targets for diagnosis, prophylactic, and therapeutic interventions. Along with this, similar work is also being carried out currently in his lab on other protozoan parasites like leishmaniasis as well. With this, on behalf of COMSTECH and His Excellency Professor Dr. Iqbal Chaudhry, I would like to thank and welcome our guest speaker for today, Professor Dr. Robello, for his lecture. Over to you, sir.
Okay, thank you very much for the invitation. It is really a pleasure to participate in this meeting. Since this network, it was my first contact with Pakistan researchers, so that is a honor for me. You have a very nice an excellent research in Pakistan, so thank you very much. I will talk about a parasite, Trypanosoma cruzi, or Triclosan. I will say Trypanosoma cruzi, I say that it's in Spanish, that is really similar to Leishmania, that probably is the most known for you. It's a kinetoplastic parasite, it's a trypanosomatid, so it has a kinetoplast, it has one nucleus clearly delimited, etc. But instead of Leishmania, it produced Chagas disease. Chagas disease is a disease that occasionates a cardiomyopathy and also some dilated pathologies in the colon, esophagus, and other organs. It's a it is a chronic disease, and it is transmitted by an insect that is called that is a triatom, and triatoms exist in America. So that's why it is an American trypanosomiasis. But also it can be transmitted vertically, transplacental, and by transfusion. So due to migration, now the Chagas disease is distributed around the world, mainly in Europe because of the migration from South America to some countries, Spain, France, and other countries. There are several cases of Chagas disease there. Okay, so I will talk about Trypanosoma cruzi, and all of the work will be oriented to genomics. What we have done with the genome analysis of Trypanosoma cruzi. I will talk about this first point: if Triclosan constitutes a unique species, to the architecture of its nuclear genome, and how can we use the information from the genome to study the mode of action, resistant mode of drugs. Okay, so these 3 points will be developed in this talk. All of the work has, or most of the work I will show, has been done using short read sequencing. This is an Illumina technology for high throughput sequencing, and these two technologies, PacBio and Nanopore, that sequence long reads. Okay, so until the emergence of this next generation sequencing, the methods of sequencing, the method for sequencing DNA and for solve genomes was the Sanger method. That is an excellent method, but you cannot make high throughput, and or you can sequence up to 1 kb of nucleotides per time. So it was very difficult. For example, the human genome took 13 years with hundreds of laboratories to know it in 2000, whereas today in 1 week you can have with this method the genome of a human. Okay, so that was a revolution, and of course the revolution expanded through different organisms, from humans to bacteria, passing for Leishmania, Triclosan, etc. So we sequenced several strains of Triclosan, and now we have some conclusions. This is usually, I know that several people work in Leishmania in Pakistan, and usually you can divide Leishmania in species, complexes, subgenera, and genus. So you have here, for example, the complex brasiliensis, the complex guyanensis, the complex Leishmania major, the complex Leishmania donovani, etc. So it is very well ordered, and you have this. You can divide also Leishmania in Sauroleishmania, Leishmania, and Viania. That is a really good. I mean, it is very well organized, the classification of Leishmania. But when we go to Triclosan, we have hypotheses, theories, but we cannot say if it is a unique species, Triclosan, or if it's a complex of species. Because of the title of my talk, probably you can see now, can know now that we consider that Triclosan is a complex of species. This is one of the theories that from an ancestor there is a parental 1 and a parental 2, and one progeny F1, hybrid trypanosomes, and different kind of groups named DTUs. I will talk about this later. Okay, so as you can see, it's really complicated, a really complicated map about the Triclosan. This is another classification where that is the accepted one now, that Triclosan is composed of discrete type units, DTUs. The DTUs is a way to say, I don't know if it is one species with lineages, different lineages, or 6 species or 4 species and 2 hybrid Triclosan. This is the current classification of Triclosan. It is divided in 6 DTUs, okay? DTU 1, 2, 3, and 4 are non-hybrid Triclosan, and 5 and 6 are heterozygotes and hybrids, okay? Because of the hybridization of 2 and 3, okay? So, and there are one hybrid TC5 and one hybrid TC6. That is what was accepted until now. What we had done was sequenced several genomes and analyzed in detail the maxi circle of Triclosan. It represents the genome of the mitochondria. It is the same in Leishmania. It is the same in the African trypanosomes. I think that someone has a question. Is us? No? Okay. If you don't understand my English, or if you want to ask, You can interrupt me, don't worry. Okay. When we started this work, we have the length of the maxi circle was defined as 22 kilobase pairs. Okay, this is a typical maxi circle of Triclosan with a coding region that is the same for all of the trypanosomatids. So Leishmania has the same coding region with these genes that most of them belongs to the respiratory chain. So it is like the mitochondrial DNA, our mitochondrial DNA, but also has a repetitive region and some conserved domains. This is the structure, this was the structure of maxi circles, and the length was 22 kilobases. Okay, when we sequence our strains, we have found in this map this density indicates that there is a repeat. This is a sequence of the maxi circle that we have done, and we have found here a repetition. Density means that there are tandem of repeats because it's a comparison between the genome of one strain against itself, okay? And there was another repeat inside the maxi circle. All of them, the length of all of them was 25 kb. So by long read sequencing, we could see that really the maxi circle is longer than 22 kilobases. In fact, this is a structure that we have defined for the maxi circle of Trypanosoma cruzi that is composed by the known coding region, a short repeat, and a very long repeat. And this is true for all the DTUs, okay? 1, 2, 3, all have the coding region, the short repeat, the long repeat, and an 80 rich region here. So this was one finding thanks to the long read sequencing. This is the same. This is DTU 1, 2, 3, 4, 5, and 6. And here you can see the coding region, the short repeat, and the long repeat. What I am showing here is the GC, or in pink or red, and in blue or violet, the AT content. As you can see, the AT content, for example, let's see the AT content, it's common to all of them in the coding region, changes in the short and long repeats, yes? And this is the same for all of the parasites, of the Trypanosoma cruzi parasite. Also, there are some differences inside the coding region. I will not talk about in detail of this. And this is the long repeat. In the long repeat, what we could find was this sequence that is palindromic, and this sequence is conserved along all of the DTUs. So what we can say is that this is a common organization of the maxi circle of Triclosan. Also, we saw that in Leishmania and T-Brucei, they have the same organization. Yes. And after that, what we have done was clustering of all of the sequences of maxi circles, and the clustering of maxi circles show that they can be clustered in 3 groups, A, B, and C, meaning that Triclosan have 3 types of maxi circles, A, B, and C. Only 3 types. Those types, that result means that probably the origin, there are at least 3 groups at the origin of Triclosan. So this result gives clues about the origin of Triclosan. So probably there are 3 at least ancestors from Triclosan. This is a map showing this is a mitochondrial a phylogenetic tree from mitochondrial sequences, where you can see here those who has the maxi circle A, the maxi circle B, and the maxicircle C. Yes, this is TC1. Here is TC3 and TC4. And here is TC2. These are the names of DTUs. So since we saw that there was not enough information to make phylogenetic inferences from the trees, what we have done was to take nuclear genes and make the same phylogenetic analysis. Here we have used 85 genes for each strain, and you can clearly see that instead of 3, there are 4 groups of Triclosan: A, B, C, and D. These 4 groups of Triclosan have particular maxicircles. The clade A has the maxicircle A, the clade B, the maxicircle B, the clade C, the maxicircle C, but the clade D has the maxicircle B. And hybrid trypanosomes, this is one hybrid Trypanosoma cruzi, this is 6, this is another one. All of them, and this is TC5 that is also hybrid, have the maxicircle C. When we use more than 1,000 nuclear genes, we have the same results. Triclosan is composed of 4 lineages, 4 clades, clearly separated. So we proposed a new classification. Instead of this classification, where, for example, TC4 could not be located in the organization evolutionary story of Triclosan, we have found that the Trypanosoma cruzi complex, because we can clearly say that this is not a unique species. The Trypanosoma cruzi complex is composed of group 1 and group 2. Group 1 composed by A, B, and D clades, and group 2 composed of clade C. The uppercase means the clade or lineage, and the lowercase means the kind of maxicircle it has. And we have found only one hybrid. So the previously named or currently named TC5 and TC6 in our hands or our results say that there is only one hybrid kind of Trypanosoma cruzi that we call BCB, because it is composed of lineage B and lineage C, that is BC, but they have all of them the maxicircle B. So this is a new classification that we think that it can be important in concerning a study of new drugs. For example, if I develop a new drug, what should I do? Should I check against all of the DTUs or all of the groups or lineages, where at least one member of each group, 1 and 2, should be tested? Ideally, lineage A, that is more separated than the rest, and lineage C. Or you can also use lineage A and C, A and the hybrid that has the genotypes B and C. And this also should be applied to studies in diagnostic or vaccines, for example. So this is one example of how we can use genome studies for future drug development or diagnostic development or vaccines. Okay, that is the first part that I wanted to show you. The second one was the architecture of the nuclear genome of Triclosan. When we started with this work, the genome, the last genome that was published was from 2005, and it was done by a very big consortium of about 50 authors because of the difficulty to make genomics by the Sanger method. What we have done was, as I told you, use PacBio and Nanopore, this technology for long-read sequencing, complemented with Illumina. and sequenced all of the lineage that we defined. We have defined. Okay, so we have this. All of these are the name of strains. DM28C is one strain of Triclosan, or one isolate. TCC is another one. I will show some result with DM28C and TCC that correspond to lineage A and the hybrid. This is DM28C is A, and this is TCC belongs to this hybrid lineage. Okay, we sequenced several of these genomes, and the first point is that we could expand or know the real length of the genome. This schematic view, what is showing is, this is our genome, the genome that we have sequenced, and this is the old genome. So we can uncollapse some regions because, for example, here there were repeats and repeats of that cannot be solved by short-read sequencing. So in the old genome, this region seemed shorter, and now we have found that there is a tandem of genes. Yes, these red lines means homology. There are a tandem of genes, yes, in this region. So you can now know the real length of the genome. This is another of our sequences. This represents part of a chromosome of Triclosan, where this region was expanded thanks to long read sequencing. Why? Because the reads are 10,000 lengths or 20,000 lengths, and you can pass through the region. So I can sequence in one read. I can have all these sequence, and I can say, okay, there is not only one copy of this gene, but 10 copies of the gene, 20 copies of the gene, etc. So that is why we could uncollapse repetitive regions, improve the resolution, find telomers, centromeres, improve the genome annotation, and know the repetitive sequences. The problem of Triclosan is that, well, not a problem, it's a feature of Triclosan, is that half of it, 50% of its genome is composed of repetitive sequences. That's why the genome was not so good before the long read sequencing. Okay, these are the same schematic views, and here what I show is that we can also separate haplotypes. So these are part of two homologous chromosomes. So another advantage of the sequencing is the separation of haplotypes. We generated, although our results are all in the Triclosan database, and you can find them, our strains and our sequences can be found in the Triclosan, that for people who does not work in genomics. It's a EUPASDB is a bank where all of the genomes of these parasites are deposited. So you can go there and you have a lot of tools there. It is very useful to work there. You can blast, find similar sequences, etc. But we generated our own page also, that is here, Bioinformatics. Bioinformática, well, this is, and Matías Rodríguez in particular, Fernando Álvarez, and Luisa Bernal were who did this page. So here you can clear all and select, for example, the conserved proteins and the hypothetical conserved proteins only, or non-coding RNAs, or some specific genes. Why these genes? And here a parenthesis. Among the repetitive regions of Triclosan, some of them are multi-gene families. In particular, this MASP is a family of proteins there are more than 500 genes of MASP. These mucin-like proteins are another family that there are more than 500 in all of the strains of Triclosan, and the transialidase is another protein that another kind of protein that there are more than 500 genes for them. We call them the surface multigene families because MASP, mucins, and transialidases are surface proteins. That means that they are anchored, GPI anchored in the surface of the parasite, and like Triclosan that has VSG along all the surface of the parasites, the Trypanosoma cruzi has on its surface MASP, mucins, and transialidases. Okay, and here are represented in blue sky, and orange. Okay, so we generated this color code, and the classical genes are in green. So just remember that orange, blue, and blue sky are the most important surface proteins from Triclosan, and that green are all of the genes, the common genes. For example, the enzymes, from the glycolysis, the enzyme from different metabolic pathway, etc. Okay, transcription factor, ribosomal proteins, all of them are what we call conserved and hypothetical genes. And this is the representation of one chromosome. This is a chromosome that has more than 1.4 megabases. And each of these lines in green is one gene. So this is one gene, another gene, etc. Okay? So these lines are genes. The dark green is because it's conserved, so it has a homologous in the rest of eukaryotes, or in most of the eukaryotes. For example, a classical enzyme, exokinase, for example, will be in green, in dark green, whereas hypothetical conserved is of unknown function, but is present in Leishmania, for example. And you have this. And here, this blue is a MASP gene, and this blue sky is a mucin, and this orange is transialidase. Okay? If you see this representation, a surprising result for us was that the green genes were all of them together, and the red and the orange-blue genes were all of them together. You can find some green gene here, or some red here. So what does it mean? That there is a there are compartments in the genomes. We have found that the genome of Triclosan has two clearly defined compartments. The core compartment, where conserved and hypothetical proteins are present, this is the core compartment, and the disruptive compartment, because that has transialidases, MASP, and mucin. Another protein is this one that I will not talk about it. Why we call it disruptive compartment? Because the interruption of synteny among trips. I will come back about this point later. So, note that the three components, or the four components of the, I will not talk about RHS because its function is not known. We don't know what is the function of that gene. But these three components know the number of genes that they have. For example, from transialidases, there are in this strain 1.7 thousand genes coding for these proteins. For mucins, almost 1,000 genes, and for MASP, 1.3 thousand genes. So they are really multi-gene families, and all of them are located together in the genome. That was really surprising for us, because surface proteins are the most relevant in host-parasite interaction. Okay, so this is a representation. of the compartments, and of course, they have different properties. It's like Triclosan has two genomes, a disruptive genome and the core genome. This is showing the GC content, yes, in green, the core, and in pink, the disruptive compartment, and they are very different. And you can see here also how change the GC content. Here we are representing the GC content, so we can see here the difference. Okay, this is more than the same. And also at the level of transcription, there are important differences. These are the three main stages of Triclosan: tripomastigotes, epimastigotes, anamastigotes, and the transcription. This represents density of transcription. These are reads, yes, and density of transcription when we have done RNA-seq. Here we found that in the compartment, in the core compartment, the green or core compartment, the transcription is similar between the different stages, except for some genes. This is a differential gene, but in general, in the 6 stages, they have the same amount of transcription. Whereas in the disruptive compartment, transcription goes down, and only some genes are highly transcribed, but another one are almost silent. So there is a functional correlate among these compartments in the genome. That was a very important, very relevant for us finding that Triclosan put in one place in the genome all the, let's say, essential genes needed for metabolism, etc., and in another place, all those that will contact either the insect, either the mammals, and that means that the immune system, etc. So that seems to be the strategy of Triclosan for defenses, okay? Because here they can generate some variability and without affecting this part of the genome. So gene expression is lower in the disruptive compartment. Gene expression is lower in core boundaries with disruptive compartments, okay? Here also. So they deserve to be studied. So that is another point. So as I told you, we defined the 2 compartments in the genome, and the question is, why do call it disruptive to the disruptive compartment? This is a region of the core genome of Triclosan. Each of these green rectangles represents genes. This is one gene, another gene, another gene. If I go to the trip, yes, this is the representation. This gene is this one, this gene is this one, this gene is this one. The point is that I can, if I am in the web page, I can put here my cursor and see, I will find the links to the peptide sequence, to the gene nucleotide sequences, etc. And here, this is a schematic view of synteny. This is our genome, DM28C 2018. So if you, let's see this gene, yes, has synteny with another gene of Triclosan. This Brazil is almost identical, and strain almost identical to our strain, but also with Y and CL Brenner of Triclosan. So there is synteny among Triclosan as expected, but here and here, these are 2 genes of Triclosan, and this is one gene the same gene in Leishmania. And this is for all of them. If you check the order, in Triclosan, yes, these directional clusters of genes are conserved, are the same to T. brucei and Leishmania major. We can say that the core genome of the trypanosomatids are really similar. Some difference can be found. For example, these two genes have not synteny here, and probably they are in Triclosan or T. brucei, but in another place. But in general, this is the view of a synteny representation between Triclosan, T. brucei, and Leishmania. Note that here is T. brucei, and here is T. brucei gambiense, and this is Leishmania major. So we can say that they are really similar at the genome level. But when we compare a region where half of it is core and half of it is disruptive, then you will find that the same for the green region, the core region, the same. These are the genes of our genome, yes, DM28C are here. This is the almost identical clone Brazil, and you have all the synteny relationships here. This is Triclosan also, this is CL Brenner. Now we jump to T. brucei and T. brucei gambiense, and know that they are practically the same among them. And also with Leishmania, this dispersion here is because the intergenic regions of Leishmania are longer, but this gene has an homologous and they are the same order. What does it mean? That in millions of years, these trypanosomatids conserve the order of their genes. But when you go to compare this part, that is the disruptive compartment, you find an empty region here. So this is what Triclosan has been doing during evolution, generating copies and copies and copies of the sulfate genes that are specific of Triclosan. And probably here is the main difference between Triclosan, T. brucei, and Leishmania. This is not new for trypanosomatids because T. brucei, its main difference is BSG genes. So they have developed one strategy, evolutionary strategy, and Triclosan has developed this other one. So why we call it disruptive? Because of the loss of synteny with other trypanosomatids. This region is disruptive with respect to other trypanosomatids. That was we published in the paper of the genome. Okay, but when we went after we had the genome of Triclosan, only of Triclosan, Luisa Bernard did this comparison. Here, what we show is the chromosomes on one strain, that is Triclosan esmeralda. And here is our strain. When we compare one to another chromosome, chromosome 2 has an homologous with the 39 of this strain. This 4 has an homologous with the number 40, etc. But there are some chromosomes, this one, that we could not find homologous. It's a comparison by sequencing, by homology of sequence. The same with chromosome 3, the same with chromosome 5. When we analyze another Triclosan strain bag, the same. Number 1, number 3, number 5 do not have homologous. And when we compare with Brazil, with TC, the strain Brazil, the same. Our chromosome 1, our chromosome 3, our chromosome 5, and more, do not have synteny and homology with any chromosome from from the other strain. And as we expected, why they don't have homologous? Because chromosome 1 is almost entirely disruptive. Chromosome 3 is almost entirely disruptive. This means red-blue. And chromosome 5 is almost entirely disruptive. So when we compare inside the Triclosan complex, two strains that are almost identical, they have a very big difference in their disruptive chromosomes. Yes? And on the other hand, chromosome 2, that has high homology with chromosome 1 from strain Brazil, is green. That means it's core genome, and core genome. Okay? So, the impressive result here is just a better view of comparison between Brazil and our strain in the first 4 chromosomes. Our chromosome 2 is almost identical to this one. This 4 is almost identical to the number 2, let's say, from Brazil. But here, in the number 1 and the number 3, that they are disruptive, we cannot find homology. So the impressive result was that the disruptive compartment presents large variation even intra-trypanosoma cruzi complex. That means that from an evolutionary point of view, this is a modern I don't know if modern is the correct word, but it's a modern change in evolution. Probably they are continuously changing, and instead of change one gene at a time as Trypanosoma brucei, these regions are changing stochastically, and they could select a population. They could select a population that is that can make the infection better than another one when there is an immune response, for example. So that was the point. After that, we wanted to see if there were different levels of expression in the compartments. So here you have our chromosomes, yes, this is our chromosome 1, 2, 3. And what I am representing here is another point. These boxes are genes. Each of these rectangles correspond to a gene. Green, green means low expression, and pink high expression. Gray is medium expression. So now I am moving to gene expression, RNA expression. So here you see that this chromosome, yes, has most of the genes of chromosome 1 are of low expression, and most of the genes of chromosome 2 are high expression. Chromosome 3, low expression, and chromosome 4, high expression. Okay? This is what we are representing here. And this is the stage epimastigote, and this is a stage tripomastigote. So, asterisks indicate genes that are differentially expressed. And note this, in this chromosome where most of the genes are of low expression, in tripomastigotes, these genes are upregulated in any case. So all of these asterisks means that they are more expressed in tripomastigotes, the infective stage of the parasite, than in epimastigotes. Whereas in this chromosome where most of the genes are of high expression, there are not so many differentially expressed genes. The same in the third one. Low expression, but upregulated in tripomastigotes with respect to epimastigotes. In the fourth, high expression and no significant, no very high changes in the differential expression. As you can imagine, these ones represent the disruptive chromosome. So the disruptive chromosome, the disruptive compartment have low expression, but differential expression. So we can see that it's a compartment that is specialized in the infective stage of Triclosan. And that happens always, always, always. So the organization of that genome has consequences as expected at the host pathogen interaction, host parasite interaction level, because they will overexpress genes related with the surface, okay? Whereas these represent the core genome. So there is a very different pattern of gene expression in the core. As a summary, Triclosan genes specialized in the interaction with hosts are located together in the genome, occupying in many cases the entire chromosome and forming the disruptive compartment. We call it disruptive because the synteny is interrupted, and not only between trypanosomatic, initial reason, but also between strains of Triclosan, even from the same clade. That was impressive for us, because probably during one infection, they are changed. This variability is probably the main reason for the lack of correlation between lineage and clinical symptoms. When I talk about the lineages, A, B, C, and D, several work has been done trying to say, well, this can generate cardiomyopathy, this another can generate asymptomatic Chagas disease, etc. And it could not be found a correlation. And probably it's because the great genomic plasticity of Triclosan. And finally, the core compartment behaves like a more normal genome and constitutes, in our opinion, a potential reservoir of new targets for drug development. Another point is that it is important that if someone develops a drug against leishmania that is very efficient, probably its target will be in the core compartment. So it is important to check if it will be present, it will be effective also in Triclosan, for example. So that is the second point that I wanted to show you. And very briefly, since probably I know that most of the group of Professor Samer and Iqbal, and probably the people who is in the seminar work with leishmania, not with cruci, and with related and drug development and drug mode of action, etc. It's important. I wanted to comment how we can use the information from the genome to study drugs, new drugs, mode of action, or mode of resistance. One point can be the induction of resistance. So we can induce resistance in vitro and later analyze the presence of mutation, change in gene expression, gene duplication, etc. And that could be one possibility. That is what we are doing now in a project with Professor Paul Denny and GSK. Yes, and is to produce mutants from the Chagas box developed by GSK, and to make genome and transcriptome analysis. This is a strategy. It is here we have the wild type parasites. We start to pressure them with the drug, in this case the compound number 11, and see if there are differences at the EC50 after several passages. After that, we make nanopore and Illumina. We'll show just the example of compound number 11, where the control is here. This is the control in, sorry, because the Spanish, this is growth, percent of growth. This is the control in blue. And these are two resistant clones that were selected here. So we could obtain resistant phenotype of Triclosan against this compound, and we have done genome analysis. And we found a duplication in a very big region, and so we are analyzing the genes inside this region, because one of the things that the parasite did was to duplicate this region. That means that probably here there are some genes that contribute to resistance. And another analysis is SNP, because single nucleotide polymorphisms can change the targets, the proteins that are targets of the drugs can change, and in the sense of being functional of an enzyme, for example, but not sensitive to the drug that we are analyzing. So that's what we are doing in the context of a network, trying to find new drugs or new mechanisms of resistance. So genome analysis is very important, because we can compare, and in the case of people who work in Leishmania, the comparison between natural isolates that are naturally resistant with respect to reference strength, that could be important. But also induction of resistance in the lab is an important tool to know the mode of action, because usually we find the drug, but not the drug, the mode of action. Okay, that is one approach. And the second one that I will just refer very briefly is that we have determined the number of single copy genes that are more expressed in mammals. The pipeline is and bioinformatic pipeline to generate a list of single copy genes, selection of those that are expressed differentially in amastigotes or tripomastigotes. So these are the mammal stages in Triclosan, and make CRISPR knockout experiments to determine if they are essential. If they are essential, then we can go back to people who work in chemistry, yes, and in medicinal chemistry, try to generate inhibitors. Okay, so this is the pipeline. We have our genome. We select a group of single copy genes in our genome, and go through the genome 2, that is from clade B, genome 3 from clade C, etc. Go to all of these genomes and generate a list of unique genes, of single copy genes. After that, we analyze which of them are more expressed, it is more intense red, in amastigotes and tripomastigotes. And of that, we select these genes to make a knockout by the method of CRISPR, and inducing by putting resistant genes, number 1 and 2, and we we can generate knockout and confirm if they are essential. So knockouts, what next? Knockout of annotated genes, design of inhibitors from predicted structures, and determine the potential relevance of hypothetical proteins, because there are several hypothetical proteins here. And here is where networking is important. We have found that there were 168 genes in this list. Half of them, about 80 genes are hypothetical. The other 80, we know the function. And of those, we cannot work with 80 genes. Of those, well, we start collaborations with different groups. So one group make one knockout, 5 knockouts, another 5 knockouts, etc. And we try to determine which ones are essential, and try to start to design inhibitors. And the same work can be done in Leishmania, for example, to make the same, because the pipeline was already okay. So sorry if it was so long, but that was the last. This work was done in our laboratory of host-pathogen interactions, Unidad de Biología Molecular. The work in the analysis of the genome, most of the work has been done by María Laura Chiribao, Gonzalo Grave, Matías Rodríguez and Fernando Álvarez from Facultad de Ciencias. Yes. And several work in transcription was done by Florencia Díaz. Yes. Carlos Sanz has done the work in generation of resistant genes. Sebastián Pita, single copy gene. Some collaboration already in course, and this is our group. Muchas gracias. This is she's Luisa Bernard, that has done most of the genome analysis. Gonzalo Grave doing genome analysis also. Here is Maria Laura, CRISPR experiments. Carlos Sanz working in the resistance and genome analysis, and Florencia Diaz working in genome analysis, transcriptomics, and epigenetics. Carlos Pita has been working in the pipeline of single copy genes. So thank you very much.
So thank you very much, Professor Robello, for your very informative lecture. And I'm going to the chat box to see whether we have any questions. And I can see only one question, that how we can treat the trypomoniasis.
Sorry, I will.
Professor Robello, there is a question that how we can treat the trypanosemiasis.
I understand how we can. Sorry,
how we can treat the infection due to tryposoma.
How we can detect?
No, treat. The treatment, the medicine. Yeah.
So, sorry. Well, I do not talk about the disease because I wanted to show the results, but the treatment, there are two drugs that were found in the 70s. The names are, they are nitro-derived drugs, benznidazole and nifurtimox, that have several side effects. They are efficient during the acute phase of the disease, but it's not easy to find an acute case of the disease because sometimes the symptoms are mild. So, and during the chronic phase, they cannot cure the disease. But if there is a peak, then the treatment is used. It is good, the treatment as in pediatrics. So when after congenital child disease can be treated with this drug, but they have several side effects. So treatment is not efficient. So that's why it is important to find new drugs.
Okay, thank you, Professor Robello. People are much more interested in your presentation, in your slides, and that's what I responded them, that the slides are available. Your lecture is actually available on the YouTube of the COMSTECH site. So if they are interested, so they can see from there. So once again, thank you so much, Professor Robello, for your very kind and informative lecture. And I will share your email ID with the participants if you allow. So if they
Yes,
are
of course.
more interested in any kind of knowledge or any kind of information, so they can contact you directly.
Perfect for me. It was a pleasure. Sorry if I went very fast, but well, but my message wanted to be that we are not so long. I mean, we must try to collaborate, to have more collaboration between groups working in medicinal chemistry and genomics, because people who work in molecular biology and genomics, that's my case, are always thinking how to contribute to the development of new drugs. And I don't know how to make new drugs, but you know, biochemistry know about enzymes, and other people knows about chemistry. So that's why I wanted to show that we need to make more connections about this. And of course, that if some people is interested in working in this orientation in Leishmania, that could be fantastic. Try to have sequences of Leishmania and try to be the mode of resistance, etc.
Yes, Professor Robello, I totally agree that the collaboration always ended up in new findings, which found to be more effective as compared to the individual research. Yeah. So thank you so much, Professor Robello, and then
Okay.
see you at another time, maybe in
Yes,
physical.
I hope. Thank you very much, and thank you to all the people. That are in the talk. Okay.
Yeah. Okay.
Okay.
See you. Bye.
See you. Bye bye.
Have a good day.
And thank you for the comments. I tried to read the comments. Thank you very much for the comment. Okay, my mail will be available. So someone asked, how can we collaborate? So you can write me and we can. Okay.
Yeah. See you. See you. Thank you.
Bye bye. Thank you.
Bye bye.